Thank you guys so much. I would like to now welcome Dr. Hayflick up for the NBIA research overview.
Thank you very much. Could I have my slides up, please? I am not stepping down. It's been a real honor for me to work all these years with Patty, and it's going to be an honor for me to continue without missing a beat. So my first slide will be a bunch of logos. Oh, no, it'll be Oregon, where there's verdant hills, no wildfires right now, beautiful sky, plenty of water. So my only disclosure is that I do have an association with the Spoonbill Foundation. I'm no longer in a leadership role. I'm actually the cashier and recorder. And if you've ever seen my handwriting, you'll know how much trouble that is. So the Spoonbill Foundation, for those of you who don't know, it's a not-for-profit organization that we formed, and the intent is to bring therapeutics forward in partnership with the community at the lowest possible cost and as swiftly as possible. And even though you all aren't exactly part of the Spoonbill Foundation, we consider it to be very much of a community collaboration. I wanted to, I know that many of you know the story, but I wanted to tell those who don't know the story kind of how I got started in 1991, right before some of you were born, maybe a lot of you were born. I had my first job. I was just out of my training, and I took my first job in Western New York, and I met a family, an Amish family, which is why I have the horse and buggy sign up here. And that family had 4 daughters, and 3 of them had a disease we used to call Hallervorden-Spatz syndrome, and now we call pecan. But we didn't know that then. And this family had a very deep and lasting impact on me. They touched me very deeply because of the poignancy of what they were challenged with 3 very sick daughters living without electricity or out in rural parts of the state. But they were managing rather beautifully with their community support, and I used to go down and visit them and help in any ways that I could. They were the family that got me started, and it was their DNA that allowed us to ultimately find the pantothenate kinase 2 gene as the causative gene for pecan. That family we've stayed in contact with the family. The family is obviously in the Amish community, very, very connected to their community, and we had the real honor of enrolling a number of Amish children in the COASY trial, and so many of my team members had a chance to work with them, and I consider that a coming full circle of sorts. The other point I wanted to start with is this is the photo of that first meeting that Patty was making reference to. And it's a very small group, but I've circled, actually that's Patty, I think, in the middle there, and there's several other people in that circle that I wanted to just call out today. So when we take research samples, we almost always consent you. We always consent you if we're going to do anything on, for scientific research on you and your families, but we needed to get a DNA sample from Patty and the rest of her family members in a way that was kind of sort of secretive, and we don't do that. We aren't doing that to anybody else. But so we had to like go to the bar on the first night of this meeting, and they were all sitting at the bar, and we plucked a hair from each of them, sort of over the course of about an hour, and none of them noticed. And you've seen us actually taking these other samples and sending them back to Oregon, and we sent those plucked hair samples back to Oregon on, I think it was Thursday night, and we actually got a result. So the question we were asking is, what is it about these sisters and cousins that makes them so generous to this community? Really, it's a pretty remarkable family, isn't it? They have been here at almost every one of the family meetings. And so we sent the samples back, and we did their genomes, right? Because we wanted to understand this generosity that really you don't find in the average person. Or maybe you do, and we just don't see it in each other. And so I'm happy to say that we found a gene duplication for what's called the generosity gene. We all have it, but we usually only have a single copy of it. And we found that actually in Diane and in Vicky and in Regina, and not surprisingly, it's in Patty also, they have a 7-fold duplication of this gene. Anyway, it explains a lot. So before I speak to you each time we meet together, I do a literature search of the entire biomedical literature. For our disease set, it doesn't take that long, about 4 seconds. And this is a timeline across the bottom. It started I think in 1993, sorry, I can't see that, wherever it started. I searched on NBIA, because that's sort of the capture. There are papers in our field that don't use the term NBIA, and so they're not captured. But I wanted to just show you over time what's happened with the publications in our field, and that the name was invented by somebody in 1996. I think it was me. And then the papers started to accumulate, and you can see we were having, on average, about 50 papers a year. And then you'll notice towards the end that things start to tail off. And in case any of you have forgotten, I wanted to just draw attention to the fact that there was a decrease in publications because there was a decrease in work going on. Many of us had to not only stay at home, but actually our experiments that we had in process, our animal colonies, our human cells, everything we were working with, that sort of the momentum that we establish in our labs, we had to quiet that down, shut that down for many laboratories. And so there was a sort of a slowing for many scientists. But I wanted to remind you that there was a pandemic, because it's easy to forget. And I wanted not to miss the opportunity to tell you that the reason we're sitting here today is, there's 2 reasons, both science. One is evolution of the virus, right? The virus doesn't actually want to kill you. What it wants to do is it wants to infect you in a way that you can infect as many people as possible, because that's a selection process. That helps the virus live on. If it kills everybody that it affects, then it sort of stops there. So the virus has evolved. That's evolution/science. But the other reason many of us are here, though many of us did lose family members and loved ones from this terrible disease, is that mRNA vaccines came along, and they came along at an astonishing rate because a lot of research, a lot of scientific work had been done already. And the person who blazed that path was a woman scientist who was sort of sidelined as an academic. She was not well respected at the institution where she did most of her work, but she had an idea, and it was a really good idea, and she persevered, and she went on to sort of establish the kind of foundational science, and actually went on to be a part of the companies that ultimately developed the mRNA vaccines. So woman, breakthrough science, and an immigrant. So back to the science. We're about halfway through, a little bit less than halfway through 2023. I think that the papers will surge again. Much of this was gene discovery, animal models, reporting on that. We now have pretty decent models in many of the NBIA disorders, not all of them, but in many of the NBIA disorders. And so now really where we're getting some momentum is around ideas for therapeutics, the resources we need to test promising therapeutics, and it's onward from here. So what happened when I put that information up on that slide? What did you do when I put that information up on that slide? your eyes went to the disease that is affecting your family. You saw BPAN, you skipped over it. You saw PLAN, you skipped over it. You went right to PCAN. Or you saw BPAN, PLAN, oh, you stopped at PLAN. Or you went right to BPAN. Because those are the diseases that affect your family, and that's what you want to know about. But I'm here to tell you, you also want to know about what's going on in all of the other diseases, not to have something to talk about with the families at your table, but because what's happening in the other NBIA disorders is important for what's happening in the disorder that affects your family. I think it's really important that we're here together, and I think it's really important that the disease groups cross-pollinate. I think that that's exceptionally important to our science. We now know, with the gene discoveries and some basic understanding of the biology of many of them, not all of them, we know that there's a lot of intersection around PCAN, COPAN, FON, and MEPAN. Probably PLAN should be up there as well. Maybe MPAN, but we don't really know the key information about the MPAN protein function. What we do know is that MPAN, PLAN, and BPAN are in this continuum of protein aggregation in the brain, with MPAN being a synuclein-dominant disease and BPAN being a tau-dominant disease. And so our knowledge in these disorders of what comes first, why these proteins accumulate, how they lead to accumulation of other proteins and dysfunction of the cells is going to be very, very important to understanding their biology and thinking about therapeutic interventions. They will teach us about each other. And I want to remind particularly the newer BPAN families, before there was a BPAN, before 2012, the PCAN families fundraised in order to find a new gene. So somebody else would have eventually found it anyway, but we found the BPAN gene in 2012 with funds that were raised by the NBIA Disorders Association and largely by fundraising from other families. So I think it's really important that we're here together. Penny and I talk about this moment of sitting at the picnic, right? It was a picnic in 2010 or 9 or somewhere in that ballpark. And it was very, very soon after that we sort of gathered the families that we knew had this particular pattern of disease and went after the BPAN gene. But it was these two families in particular that stood out to us because it was their stories and sort of the intersection of hearing their stories in a way where we were finally open to seeing, oh, that's the same disorder. We now can see that in this sea of difference, we have some common, some undiagnosed, unknown disorders, but with commonalities that could be enough to help us go after a gene. So now I'm going to leave it up a little bit longer, and you can actually first jump to your disease, but then I hopefully read. What I've done is I've delineated what I think is some of the more important work going on in each disorder. I apologize, I may have left things out. I have highlighted the things that I'm most enthusiastic or most excited about, and I'll admit to you that some of that is work going on in our own group. I get to be biased. And so in PCAN, the PCAN group heard about the COASY trial this morning, and there's quite a lot of going on in the therapeutics world in PCAN. There has been historically maybe a little bit more action there, and I'll talk about that in a bit. There's also gene therapy work going on in PCAN that some of you may not be aware of. There's gene therapy work going on in most of these disorders, and I think what we develop and learn for one will benefit the others because we're targeting brain in all of them. In the PLAN world, that's probably the disorder for which gene therapy will lead. So the work by the UCL group and Bloomsbury Therapeutics in the UK is, as far as I know, and you probably heard from Audrey Sue this morning. I didn't actually get to hear her talk. The work is chugging along and hopefully will be in clinical trials within a few years. There will be a US site. Their intent is for there to be a US site, and we have had only the beginnings of a conversation with them about having a site in the western part of the country, possibly the northwestern part of the country. In MPAN, the greatest challenge continues to be what's the protein doing? We don't understand its most fundamental role in biology, and that is really important for targeted therapeutics. Gene therapy isn't a targeted therapeutic in the sense that you have to understand the biology, you just have to know the gene. And so there may be a future for gene therapy in MPAN. I know that the London group, Dr. Kurian's group, is also working on developing models of MPAN that they can then use as a tool for testing gene therapies, right? You have to be able to put the gene into a model of MPAN and see if you're correcting the defect. We do that work in cells, in organoids, in animals before we go into humans for safety reasons, but also to see if it's having the effect we want it to have. In BPAN, there's an enormous amount of work going on. We've recently published a best practice guideline with support from the family organization, support from many of you. We are working on a PLAN, an INAD/PLAN best practice guideline, and Jenny Wilson is leading that work with Audrey Sue. We hope to have something out within the year. We'll be working with the family community and have a sort of a review process by sort of a handful of INAD families for that. The London group has a paper that is in review that I'll talk about a little bit later with some ideas for possible therapeutics. A lot of interesting work going on there, including in RNA therapy for splicing for a particular kind of mutation in BPAN, something we've talked to them about for a long time. In MePAN, it's still an ultra-rare disorder, but the MePAN community has I would say, you know how on Airbnb they have super hosts? In MePAN, they have a super champion in one parent who has driven scientists to work together, to develop resources, to share resources. He's incredible in his effectiveness, and he brings us together at regular meetings to talk with the Israelis, with the Texans, with the folks in Europe, the folks in Finland about what's going on and what we can be doing to speed along that field. I'll have a couple of slides from Dr. Venkateswaran from Ottawa on the work they've been doing on fawn. COPAN is being led by the Italian group. They've got a nice animal model, and they're working on therapeutics development as well. And then I want to remind us all that not everybody has a diagnosis. So whether you want to call it no pan or not yet pan or where's my pan, there's people who don't have a diagnosis, and their journey, you remember your diagnostic odyssey. Some people are still on it, right? We haven't forgotten you. I remember when we found the BPAN gene, and we usually return to families and tell them we've discovered that there was this mutation in your family, and we did that with an Italian family, and they wrote back with gratitude, which was wonderful, but they said, we thought you had forgotten about us. So my message to you is we never forget you. We never forget you. I also want to call out that it's I think easier to be here when your child is living, when your loved one is living. I think it's hard to come here after that person has died. And families, individuals still make that choice, and I think it's an incredibly courageous choice, and they bring this energy to the group that is essential for us. It's essential for people who are transitioning, families that are transitioning to that phase themselves, but they have this knowledge and experience, and they have an enormous contribution that they can bring. So I want to call them out respectfully. So I'm going to go in order of, sort of semi-order of gene discovery. In PCAN, we've had 3 clinical trials, deferriprone, pantethine, and retrophin trials, which none of which showed a convincing evidence of any therapeutic benefit. I don't know if there's any further studies to be done with any of those. COASY, you know about, if you were in any of the PCAN sessions, there are plans for further studies we'll be communicating directly with the pecan family community. The pantazines are in development, discovered by Dr. Jakowski's group at St. Jude and being advanced by the COA Therapeutics Company. And then we have, through funding from you all and the Italian group and the German group, we have done a gene therapy study. So we have a mouse with mutations in its pantothenate kinase 2 gene, in the pecan gene, and the mouse has a very good set of changes in the brain that are very, very specific to the same brain region in human pecan, and they're changes that we see iron changes, we see mitochondrial changes. They're very, I would say, compelling changes. We see dopamine changes, and when we give them CoAZ, you all know, they get better, those changes get better, but when we put the gene back in, we actually shoot it right into the globus pallidus, the brain also gets better. And so we're really excited about a future for gene therapy in pecan. The main reason being that we don't have to hit the whole brain. We don't have to get it into every cell throughout the brain. We only have to get it into a small targeted region, and so the viral load is much lower. And so we're talking to a variety of organizations about how to move that forward. There's some foundations that we can work with, and there's some companies that are also interested. In the pelan world, there was a discovery made in fruit flies that suggested that an FDA-approved compound might have therapeutic benefit, and some families chose to try it, this thing called dicipramine. We did it in Oregon, and the kids that we treated and we heard about had pretty unacceptable, so this is the tolerability thing, right? Unacceptable side effects that their parents were eager to stop that medication. I personally don't think there's a future for dicipramine in this disease. There was a trial by a company called Retrotope. They've never published their results. I don't know, the clinicaltrials.gov says the trial is still on, but I don't actually know if they have anything to report. We've heard anecdotally of some, I would characterize them as quite serious side effects, and so I have concerns, and I know that the company is in bankruptcy. Gene therapy for INAD, as I said, is as far ahead as any therapeutic, and the UCL and Bloomsbury Group are working together. Again, I know the INADers the families heard about this from Dr. Su and Dr. Hurd this morning. Baylor also has a partnership with INAD Cure and is working on a separate and distinct gene therapy, but I don't know the plans for that, for development and for targeted trials in the U.S. In the MPAN world, it's the same struggle. I will say that, so the MPAN was a gene that was discovered by the Polish and German group, and there's more MPAN in that part of the world than there is in the US and in many other parts of the world. And so we have ceded MPAN to them in some ways. They're leading the clinical natural history study, and hopefully we'll also be leading some clinical trials. There's some new investigators interested in MPAN and trying to do some foundational work, and so that gives me optimism. This is a slide from Dr. Venkateswaran in Ottawa about fawn. So this is an example of a clinician scientist who has embraced the role of disease champion. So when we found the fawn gene, we were doing too much already, and we didn't even have the BPAN gene at that time. So we have to kind of pick and choose a little bit because we have limited capacity, and we were never able to fully drive the fawn research program the way it needed to be driven, and she was ready to do so, has stepped in. She's partnering with other investigators around the world in a very effective way. She's built a team looking at very diverse scientific and experimental questions, just the kind of thing that needs to be done. And because she's a clinician, she's a child neurologist, she's focused on understanding enough biology to get to therapeutics. That's her goal. So I'm really, really pleased about that. So BPAN, you probably heard from Bertrand this morning that the auteur du BPAN has had a single scientific meeting about maybe a year and a half ago, and they're actually holding one very soon, a second one. This is the family group in France for BPAN, and they've reached out across Europe and the United States for participants. They've really been a very effective force, and so that's worth watching. There's also a scientific meeting coming for the NBIA disorders in Poland in October, I think, of this year, 2023. And there also will be a separate satellite meeting for scientists around BPAN. There has been what's called a landscape analysis done of the BPAN scientific landscape so that the community understands what we know, what we don't know, where the big holes are, and what we need to kind of fill in the thinking about therapeutics. That landscape analysis has not been widely shared. It's still in the final stages of its development. My expectation is that the family organization, the organizations will have access to it, and it'll be made public and available to you all. But it's a really important resource for identifying what the needs are and for helping guide scientists to filling those needs. I have some ideas myself, and I'll tell you what those are. So there's work that's been done, again, by the UCL group in developing a system for screening drugs to see if they might help in BPAN. And in order to do that, you need a system that allows you to do kind of a high throughput screening so that you don't have to go chemical by chemical. And this is it's a challenge to actually develop the assay, to develop the test itself. But it was cells from your family members that enabled this. So we had a research conference on BPAN not too long after we discovered the BPAN gene out in Oregon. Many of you came out to Oregon. We had scientific talks, we had a lot of fun, and then we had a clinic, and you gave us skin samples. We sent those samples back with Dr. Papandreou to the UCL, and he set up this test. In order for this to work, he turned your children's skin cells into brain cells, specifically the kind of brain cells that sit at the midbrain, at the Parkinson part of the brain. And then he had to find a readout from those cells, something that said, this is a BPAN cell, this is not a BPAN cell, and something that would be able to differentiate those. He did all of that work. He screened a library of 100,000 plus compounds, and he identified a handful that actually are FDA-approved drugs. They're not super non-toxic drugs. They're drugs that we're going to have to think about how on earth we're going to do that. But we're very committed to working with them and exploring the possibility of either modifying those compounds or taking some of those FDA-approved compounds forward in small trials in kids with BPAN. Penny and I have a meeting with a set of doctors at OHSU soon after we return from this meeting to talk to them about some of these hits and whether we think we could do a safe study in your children for some of these hits. So watch this space. You'll be hearing more. Their paper reporting this work is under peer review, and it's really this risk-benefit question that we're trying to weigh. I did have a communication from the head of the Polish NBIA family organization, and he informed me that there is a clinical trial underway in BPAN in Poland using compounds that I was unfamiliar with but now have learned a little bit about, called genistein compounds. There are a few papers, not a lot, there are a few papers reporting these as having some therapeutic basis in other neurodegenerative diseases, Alzheimer and Huntington, and also in cancer. The thing that caught my eye, though, was that the Alzheimer and Huntington papers were by the very same Polish group that is now studying it in BPAN. That's fine. That isn't that doesn't lessen the potential for those compounds, but it does raise a question of, are these studies being replicated by others? And we need to see kind of independent peer review of this research. I was being told of rather impressive results, but I have a healthy skepticism because it's not a placebo-controlled double-blind study, and it's hasn't undergone independent review, and those are essential if we're going to have a high level of rigor and integrity in understanding their potential. They are present in fava beans, and so if you don't have favism and you like fava beans, wouldn't be a bad thing to add to your diet. But let's wait until we have some more evidence. And then I wanted to pivot and talk a little bit about where the Rett community is. Some of you, particularly if you have girls, but maybe boys also with BPAN, you may have had a diagnosis of Rett or atypical Rett for some period of time. We actually follow the Rett literature pretty closely because it's also on the X chromosome. It's also X dominant, meaning there's more girls, but there are, plenty of boys also. The gene encodes a protein that does something very different from what the BPAN gene protein does, at least as far as we know today. It's very different. Why it's so similar in terms of the features is an interesting question we can talk about. I don't have an answer. But we follow the BPAN the Rett literature, and there's a compound that was FDA approved for Rett syndrome, and I wanted to talk to you a little bit about that. So again, the Rett protein is very different than the BPAN protein, and this therapeutic specifically targets the biology of Rett syndrome, a biologic change that we have no basis to think is also present in BPAN, but it's got us thinking that, yeah, we should look at that. So Rett may lead the way for BPAN. The Rett gene was discovered in 1999, BPAN in 2012. Those years are condensing. As you get on, it takes less time to do things as those foundations are built. But Rett has just gotten their first FDA-approved therapeutic. So the protein function is for sure known in Rett. We have some debate in the scientific community about the precise role of the BPAN protein. There are good mouse models for both of these disorders. There's actually 3 distinct mouse models for BPAN, and I think that they're complementary. There are other organism models as well. A very important experiment was done in the Rett models that showed that with an intervention, the disease development in the Rett mouse could be reversed. Not just stopped, but it could be reversed. And that was a really wow experiment, and really important for getting drug companies interested, because if you can reverse things, then you're really having really an exceptional impact. Rett is a lot more common than BPAN. Keep that in mind, because that also is relevant when you're thinking about a profit enterprise. But the reversibility was central. And this question has not been answered in the BPAN mice because we need something to try, right? Something to try to reverse it. Maybe it's putting the gene back in to see if you can reverse the changes that you're seeing. Maybe it's other targeted therapeutics. But so in this discussion of a BPAN research landscape, scientific landscape, I think one of the things to do is to explore the reversibility of the changes in the BPAN mouse. So this targeted treatment was only approved by the FDA because they had a baseline natural history study. So they'd been doing I don't know if they were doing it for 24 years, but they were doing it for a lot of years before they had a clinical trial of families plodding forward, telling us every 3, 6, 9, 12 months about the affected family member in your family. And it's work on your part. You'd rather do a million other things. You're incredibly busy. The FDA wants it. The FDA requires it for many therapeutics developments. And so this is something I implore you to find it in yourself to do the natural history studies for all of these disorders on a regular basis, as we ask, as the others ask, in order that this data set is readying the community for clinical trials. That's what you can be doing today. Really, really important work. FDA approved in 2023. Of course, you know we don't have a therapeutic yet in BPAN. I don't think it's going to be 23 years like it was in Rett, because things are accelerating, as I said. So when will it be? I don't know. I do want to make the point that the PCAN gene was found around the time of the Rett gene. PLAN was next, MPAN was next, FON was in there, but we're not talking about FON much today, and then BPAN came thereafter. I do think, as I said, things are accelerating, so you know, it took us 10 years to find the PCAN gene, it took us 2 weeks to find the BPAN gene. And so that's a lesson to us. We now have had several clinical trials in the PCAN community, and I foresee, and I think others would agree, trials coming in the PLAN community very soon, and hopefully also coming in the BPAN community. I didn't want to make any promises here, but I think it's, I think 10 years is a reasonable time frame. I would hope it's much sooner. And those may be not gene therapy trials, but maybe they're trials of the UK FDA approved compounds. Again, there's a lot of work to be done before we get to that stage. For MEPAN, COPAN, and the others, we don't have trials teed up, but I would say that as the work goes on in these other disorders, it will inform our work on those disorders as well. So I've talked about accelerating research. I want to make this point because it's the cold hard truth. We've talked about natural history studies and their importance. I'm not going to bang on your heads anymore about that one. The gene therapies, I think, are coming along nicely for 3 disorders. That's PCAN, PLAN, and BPAN. I think there's work now being done in MPAN from the UCL group. New therapeutic ideas, I've talked about those a little bit. We have 2 best practice guidelines, one for PCAN, one for BPAN. We're in the works with one for PLAN, so that'll be out soon. How do we make this happen faster? You know, and the answer is more people working on it. So inviting scientists to this meeting and sitting with them and talking to them and hooking them. I think Dr. Ben Mamoun is hooked. Dr. Jakowski was hooked before I was even in the field. So getting scientists interested and sort of connected to us, that's one way. And the other way is funding. If you put out a grant call and it's in the intersection of the work that somebody's doing, or you want to sort of lure somebody to the field who's not currently working in that disease, funding is necessary. And I know that, you know, many of you excel in your fundraising efforts, but more money makes science go faster. This will embarrass her, but tough. Did she leave the room? Okay. Allison, I don't need to tell you, Allison is a gem. She's so Penny, Allison, me, Allison, really the whole team, there's not a single person who doesn't carry their load in the group, but Allison is the front line for really all contact with us, research or otherwise. She is also, so she's a global resource. She's also incredibly knowledgeable about all of the NBIA disorders. I would say she knows as much about them as any other human on the planet. She's also a research leader. She brings her ideas to the subject. She leads research. She writes. She's a first author on many of our publications. She writes beautifully. She is going up for promotion to associate professor this year. She's an exceptional partner. She's also an advocate for you. You know that. And she's been doing this service for 20 years. How Allison does that is it's piggybacked on to everything else that is funded. There isn't a single line of support for Allison to do all of the care that she gives to the community. And I think as this organization advances and grows and the needs expand and it maybe even exceed what Allison, what our team is able to do from a frontline service standpoint, it's worth thinking about how to fund expertise in that way. When you see Allison, give her a hug. We have seen the picture, many of you. We have an awesome team. Not everybody on our team is in this picture, but we have really an exceptional team and couldn't do it without, everybody brings their own unique skills to the work. And then I've started talking about this a little bit more consistently, even though I'm not stepping down or have any plans to step down. I'm mortal, and we're always planning for sort of what to do with our program when we no longer are active. And so we think about this, we talk to others, we have begun a process of understanding the extent of our own rich research resources and have been thinking very specifically and deliberately about how to distribute those, how to manage those in a way that they stay valuable to the research community. And all of the samples your families have given, we have sent literally hundreds of tubes of blood and skin biopsies back to Oregon. That's a repository that we share with others in the community. We, as within the limits of what we're able to do from a regulatory standpoint, we share data. That's fundamental to the work that we do. So we're here together because the science makes sense for us to be here together. The clinical care certainly does. The expertise in dystonia is good for taking care of almost any NBIA disorder, including not yet PAN. There's certainly strength in numbers, and this organization is phenomenal. And so, you know, if the BPANers go off and do their own thing, this organization loses momentum, and you have to start anew. And so I think there's enormous value in us staying together. That's a point I make frequently. This is not us entirely. This is from a past meeting, but it's the best I had. We have strength in numbers. We're here as a important group, and I think it's very important that we stay that way. Thank you.
For those of you who've been getting Amber Alerts about the chemical fire, we talked with the Office of Emergency Management, and it's on their website that you only have to shelter in place if you're within 1.5 miles of it, and the picnic site is 3.4 miles away, so we are safe to go to the picnic and be outside. Yes, the hotel's actually 2 miles away, so we're better to go. So anyway, I want to read you some things this morning if you weren't all here, and about, let's see, if you were driving, parking is on the residential side of the street only, and there's a wheelchair ramp directly in front of the recreational center for those that need it. There's air conditioning indoor area, and we have an outside, so whatever is your preference. And there's large, spacious, wheelchair accessible bathrooms. And the buses are going to be right at the front entrance. They're going to start loading at 12:15, and we're going to take off at 12:30 for the picnic. So see you all there, and have your NBIA shirts if you have them.